Hello everyone, welcome back. Today we will discuss an important topic in molecular biology, that is regulation of gene expression. How a gene expresses itself. It expresses itself by two ways. One is formation of mRNA from DNA, that is transcription, and finally the formation of polypeptide from that mRNA by the process of translation. So there are these two steps involved in gene expression. In this lecture we will discuss about the Operon concept in detail. What is Operon and who gave this concept of Operon, and what is the composition of this Operon. So let's start. Firstly, Operon concept. So the first study on gene expression was done by two scientists named as Jacob and Monod. So they studied the regulation of gene expression in detail in 1961. There were two scientists. इन दोनों scientists ने regulation of gene expression पर study किया है and proposed a model called Operon model to explain the genetic basis of enzyme induction and repression. And they explained that how a gene is responsible for enzyme induction and repression. Operon are the segment of genetic material, that is DNA. Basically जो operon है वो DNA के segments होते हैं that functions as regulated unit that can be switched on or off. और ये act करते हैं as a regulated unit जिसको कोई switch off भी कहता है, on भी कहता है. That we'll discuss in upcoming slides how it is done. So this is basically a segment of DNA which consists of various genes. Firstly, there is the control region which consists of two genes, that is promoter region and the operator region. Then next comes the structural genes. इसके अलावा इसमें एक और gene present होती है represented by I. This is known as regulatory gene, and this gene is responsible for the formation of a protein known as repressor. ये जो जीन है ये कोड करती है एक प्रोटीन के लिए जिसको रिप्रेसर कहा जाता है. एंड दिस रिप्रेसर अटैचेस इटसेल्फ टू दिस ऑपरेटर रीजन, और जब ये ऑपरेटर पे खुद को अटैच करता है सो जो ऑपरेटर है ये स्विच ऑफ हो जाता है. एंड आफ्टर दैट ये जो स्ट्रक्चरल जीन्स है दे आर नॉट एबल टू एक्सप्रेस. तो यहाँ पे जो ट्रांसक्रिप्शन का प्रोसेस है दैट गेट्स हॉल्टेड और ये ट्रांसक्रिप्शन नहीं हो पाता है जिससे जीन्स अपने आप को एक्सप्रेस नहीं कर पाती हैं. व्हेन I'm talking about this Lac Operon that we will discuss in this lecture. So if there is presence of that lactose in the solution, then this lactose binds itself to the repressor and deactivate this repressor. और जो repressor ये फिर bind नहीं कर पाता है operator region पे, then it becomes it remains switched on. और जब switch on रहता है तो ये जो genes हैं ये खुद को express करती हैं, and after that there is a formation of certain enzymes which are involved in lactose metabolism. So that we'll discuss in detail. Operon is regarded as a complete unit of genes regulation comprising of various genes, like we have structural genes, then operator gene, regulator genes and promoter region. So ये components हैं एक operon के. Next, firstly, what is regulator gene? This gene is involved in control of activity of operator gene. जो operator gene है उसकी activity को ये regulator gene control करता है by producing protein known as repressor, और ये inhibitor की तरह काम करता है. So regulator gene इस inhibitor के लिए कोड करती है जिसे कहा जाता है repressor, and function of this gene is affected by presence and absence of a substrate. So जो substrate है, for example we have lactose as a substrate. So जो lactose है इसकी presence और absence अफेक्ट करती है इस रिप्रेसर को. इन द एब्सेंस ऑफ सबस्ट्रेट, जब सबस्ट्रेट एब्सेंट है, द रेगुलेटर जीन सिंथेसाइज अ रिप्रेसर प्रोटीन. तब ये एक रिप्रेसर प्रोटीन के लिए कोड करता है विच बाइंड्स इटसेल्फ टू द ऑपरेटर साइड, और ये जो रिप्रेसर है ये खुद को बाइंड करता है ऑपरेटर साइड में एंड ब्लॉक्स द ट्रांसक्रिप्शन, जिससे कि जो ट्रांसक्रिप्शन का प्रोसेस है दैट गेट्स हॉल्टेड. सो बेसिकली this is the regulator gene, और ये जो regulator gene है it expresses itself by the formation of protein जिसे कहा जाता है repressor protein when there is no substrate. जब यहाँ पे substrate नहीं है, for example we have lactose, जब lactose यहाँ पे नहीं है then this repressor gets bind to the operator region, और जैसे ही operator पे bind करेगा ये जो पूरा operon है gene system है it gets switched off. In the presence of substrate, when substrate is present, this regulatory gene forms a repressor protein which forms a complex with substrate. जो ये repressor है ये combine करता है substrate के साथ. For example this is lactose and it forms the complex, that is repressor or substrate complex, so that ऑपरेटर इज नॉट ब्लॉक्ड एंड ट्रांसक्रिप्शन अकर्स, जिसकी वजह से जो ये रिप्रेसर है ये बाइंड नहीं कर पाता है ऑपरेटर रीजन पे, और जिससे ये जो स्ट्रक्चरल जीन्स हैं दे एक्सप्रेस बाय द फॉर्मेशन ऑफ mRNA and finally they will form the polypeptide. Means this gene will express itself. The genes will express itself. The functioning of operon is stopped when operator gene is covered. जो functioning है operator gene की वो रुक जाती है when this operator is covered with this repressor. The next comes promoter gene. This gene provides a point of attachment to the RNA polymerase. Promoter region पे जो RNA polymerase है वो खुद को bind करता है, which helps in formation of mRNA, जो mRNA बनाने में help करता है, and is required for the transcription of structural genes. Or this enzyme is required for the transcription of these structural genes. Next comes structural genes. These are the genes which transcribe mRNA for polypeptide synthesis. So जो ये genes हैं ये transcribe होती है to form mRNA and finally to the proteins. For example, these are involved in formation of enzymes in case of lactose, Lac Operon. Next, we have this slide which shows the Lac Operon. This Lac Operon consists of regulatory sequences designated as I gene. This I gene which codes for a protein known as repressor protein. और ये जो रिप्रेसर प्रोटीन है ये बाइंड करती है खुद को इन द ऑपरेटर रीजन इन द एब्सेंस ऑफ सबस्ट्रेट मान लीजिए यहाँ पे लैक्टोज है, एंड व्हेन देयर इज नो लैक्टोज देन दिस रिप्रेसर प्रोटीन बाइंड्स इटसेल्फ टू द ऑपरेटर रीजन एंड दिस ऑपरेटर एक्ट एज अ स्विच एंड इट गेट्स स्विच्ड ऑफ. जब ये रिप्रेसर प्रोटीन ऑपरेटर पे बाइंड करता है तो ये जो ऑपरन है वो स्विच ऑफ हो जाता है एंड दीज 3 स्ट्रक्चरल जीन्स आर नॉट एबल टू एक्सप्रेस इटसेल्फ. Means transcription रुक जाती है, transcription gets stopped. But when the lactose is present, जब lactose present है, then this lactose combined with the repressor protein and forms a complex. और ये जो complex है it gets deactivated and it is unable to bind with operator region. और ये जो complex है ये operator region bind नहीं कर पाता है. Because of that जो operator region है ये switch on रहता है and जो structural genes है they gets expressed or transcribed. And they will form the respective enzymes. For example, this Z gene which codes for enzyme which is known as beta galactosidase, and Y gene which codes for beta galactoside permeases. Permeases are the enzymes जो कि permeability को increase करते हैं lactose के in alkali. Then we have third gene that is codes for beta galactosidase transacetylase. So जो ये 3 structural genes हैं ये 3 different enzymes के लिए code करती हैं, and this formation of enzymes is controlled by this repressor protein and this lactose which is a substrate. So in next slide we have one.
The expression of genes can be transcriptionally regulated. Such regulation is a critical feature of both eukaryotic and prokaryotic organisms. One of the classical systems used to investigate transcriptional regulation was originally investigated by Jacob, Monod, and Pardee. Their studies involved the transcriptional regulation of the genes used to ferment lactose in the bacterium Escherichia coli. 3 genes clustered together on the chromosome are required for the bacterium to utilize lactose. These were determined to be the LAC-Z gene that codes for the enzyme beta-galactosidase, the LAC-Y gene that codes for a lactose permease, and the LAC-A gene that codes for the enzyme galactoside transacetylase. The LAC-P region is the promoter that is needed to transcribe the LAC-Z, LAC-Y, and LAC-A genes as a single polycistronic mRNA. The LAC-O region is an operator site that is involved in the transcriptional regulation of the Lac Operon. The unit consisting of the Lac promoter, Lac operator, and the Lac-Z, Lac-Y, and Lac-A genes is called the Lac Operon. Located near but not in tandem with the Lac region is the Lac-I regulatory gene that codes for an mRNA that is translated to produce a protein referred to as the Lac repressor. In the absence of lactose inside the cell, the Lac repressor protein is active. In its active state, the Lac repressor recognizes and binds to the Lac operator site. When the Lac repressor is bound to the Lac operator site, RNA polymerase is prohibited from recognizing the Lac promoter, and the Lac Z, Lac Y, and Lac A genes are not transcribed. When lactose enters the cell, a small amount of it is converted to allolactose via beta-galactosidase. Allolactose binds to the Lac repressor. This causes a conformational change in the Lac repressor protein that prevents it from binding to the Lac operator site. Inactive Lac repressor is unable to bind to the Lac operator site. Without an active Lac repressor bound to the Lac operator site, RNA polymerase is able to bind to the promoter, and a polycistronic messenger RNA is transcribed that includes the Lac Z, Lac Y, and Lac A genes. This polycistronic messenger RNA is then translated to produce the beta-galactosidase, lactose permease, and galactoside transacetylase proteins. Therefore, the induction of the Lac Operon enables the bacterium to efficiently transport lactose into the cell and metabolize it.
Next comes types of operon. Before discussing about in Lac operon, we will discuss about types of operon. There are two types of operon basically. One is inducible operon, second is the repressible operon. So the example of inducible operon is the Lac operon, that is lactose operon, and repressible operon, the example is tryptophan operon, which is an amino acid, aromatic amino acid. Firstly, what is inducible operon? If in this type of operon a substrate needs to be present, इस केस में जो substrate है, for example we have lactose, ये present होना चाहिए to induce the expression of genes, जिससे कि जो genes हैं खुद को express कर पाए, that breaks it down. For example, metabolic pathway such as lactose operon in bacteria. So this is an example of inducible operon that we have already discussed. Next is repressible operon. So it is the operon which is involved in making proteins when they are needed. There is no need to make the protein when it is already available. जब ये protein already available है तब इसकी ज़रूरत नहीं है कि ये खुद को express करे in the cell. For example, this is the anabolic, that is constructive pathway involved in tryptophan synthesis in case of bacteria. So tryptophan operon is an example of repressible operon. And this operon is involved in formation of this amino acid which is involved in protein synthesis. और जब proteins present नहीं है cell में तभी ये amino acid बनेगा, otherwise this operon will be switched off. So firstly we'll discuss about the inducible operon, that is the lactose operon, abbreviated as Lac operon. So the Lac operon is an inducible operon consisting of three structural genes known as of non-lengths, designated as Z, Y, and A, which respectively codes for beta-galactosidase, permeases, and transacetylase. This Lac operon is governed by a region which is not a promoter region and an operator region located upstream from the structural genes. So जो ये region है, that is the control region which consists of these two segments, that is promoter and operator region, और ये present होता है upstream from the structural genes. जो structural genes हैं उनके upstream में present होता है. In promoter region, RNA polymerase binds and initiate transcription, while operator is an area where the repressor binds and to halt transcription. So जो promoter region है यहाँ पे RNA polymerase खुद को bind करता है to start transcription, and in operator region this repressor which is coded by this I gene, Lac I gene, that codes for repressor protein, this binds to the operator region and makes it and halt the process of transcription, means stops the process of transcription. So Lac I, that is the regulatory gene, it is located upstream from the Lac operon. ये present होता है Lac operon के upstream, that we can see here. इसके ऊपर present है ये upstream. Lac I, that is regulatory gene, codes for the repressor protein that we have already discussed. The repressor binds to the operator and in so doing prevents RNA polymerase from associating with the promoter of the operon. So ये prevent करता है RNA polymerase की binding को promoter के साथ, if this repressor is binded with the operator region, जिसकी वजह से जो transcription process है वो stop हो जाता है. This image again shows the inducible operon. So it consists of different genes. First is the regulatory gene. It codes for a protein which is known as a repressor, which is active in the absence of substrate, that is lactose. अगर lactose present नहीं है तो ये जो repressor है ये active रहेगा and it binds itself to the operator region. So जो promoter or operator region है, these forms the control region of the operon. In the absence of substrate that is lactose, ये प्रोटीन बाइंड करता है ऑपरेटर रीजन पे, जिसकी वजह से जो स्ट्रक्चरल जीन्स हैं वो खुद को एक्सप्रेस नहीं कर पाती हैं एंड द ट्रांसक्रिप्शन गेट्स हॉल्टेड. व्हाई? बिकॉज जो रिप्रेसर है जब ये ऑपरेटर पे बाइंड करता है, सो इट प्रिवेंट द अटैचमेंट ऑफ दिस आरएनए पॉलीमरेज टू प्रोमोटर रीजन, जिसकी वजह से ट्रांसक्रिप्शन. Gene regulation can be negative or positive. In case of negative regulation of gene, the repressor, which is synthesized by Lac I gene, also known as regulatory gene, that binds to the operator and halts the process of transcription and stops the process of transcription. In positive regulation of gene regulation, an activator that interacts with RNA polymerase or starts the process of transcription. So activator plays an important role in case of positive regulation of gene. This Lac operon, that is lactose operon, is an example of negative regulation, that is gene negatively regulated gene expression. For example, in E. coli, which is growing in a medium containing lactose only, the carbon source, some lactose is converted into its active form, that is allolactose. Before it is broken down into its monomeric subunits, that is lactose and glucose, this lactose is firstly converted into allolactose. It acts as an inducer. Presence of glucose, allolactose is not produced. In the presence of glucose, when glucose is already present inside the E. coli cell, then this allolactose is not produced. Inducer will stop the activity of repressor. This inducer, that is allolactose, it binds to the repressor, which is coded by Lac I gene or regulatory gene. It binds with this repressor and stops its activity, and it is unable to bind to the operator gene. The presence of lactose induces the expression of Lac operon, so that's why this Lac operon is known as inducible operon, because here this lactose acts as an inducer. So that's why the Lac operon is known as inducible operon. This image shows the E. coli cell. Inside this E. coli cell, there is no glucose, or there is very less amount of glucose inside the cell. Then this lactose is firstly converted into allolactose, which is an active form. Then with the help of these enzyme, which is known as permease, that allows the entry of lactose inside the cell. So this enzyme allows the intake of this lactose. It is then acted upon by the enzyme beta galactosidase, और ये enzyme इसको तोड़ता है इसके monomeric units में, that is glucose and galactose. So in the presence of inducer, when inducer is present, that is lactose, this lactose binds with this protein, which is previously known as repressor, coded by this Lac I gene, that is regulatory gene, and it forms this protein which is known as repressor. Then it forms a complex with this repressor, that is lactose repressor complex. Then after that, this repressor that is inactivated, it is unable to bind at the operator side. In operator side, it is unable to bind. It is not able to bind on the operator side. Then this RNA polymerase is able to bind on the promoter region. This is the promoter region, and starts the process of transcription. And after that, it forms this mRNA, which is a functional mRNA, and that codes for three enzymes, that is beta galactosidase. There are three structural genes, that is Z5A, that codes for three enzymes. One is beta galactosidase, then permease, and transacetylase. So in the presence of lactose, which acts as an inducer, these structural genes undergo the process of transcription and translation. So that's why this lactose operon is known as inducible operon. Here, this lactose inactivates the repressor, and it is unable to bind on the operator gene. Next, catabolic repression. If we culture E. coli cells in a medium which is rich in glucose, then there is suppression in the formation of this enzyme, that is beta galactosidase. ये enzyme नहीं बनता है. Even if lactose is added in the medium, अगर हम lactose को भी medium में add करते हैं, because this glucose has inhibitory effect on the formation of this enzyme, that is beta galactosidase. Another phenomenon which is very common in bacteria is suppression of aerobic respiration. and electron transport by high glucose concentrations even in the presence of plenty of oxygen, ample oxygen. अगर ऑक्सीजन कंसंट्रेशन काफ़ी ज़्यादा भी है तब भी जो एरोबिक रेस्पिरेशन है और इलेक्ट्रॉन ट्रांसपोर्ट चेन है बैक्टीरिया में सप्रेस हो जाता है ड्यू टू प्रेजेंस ऑफ ग्लूकोज. In the case of catabolic repression by glucose in bacteria, it appears that glucose affects the amount of cyclic AMP, that is CAMP present in the cells. So जो glucose है उसका main effect पड़ता है concentration or amount of cyclic AMP पे. When the concentration of glucose is high, the concentration of cyclic AMP is low, and low level of glucose are accompanied by high concentration of cyclic AMP. So it works as vice versa. If glucose is high, then cyclic AMP is low. If cyclic AMP is high, then that means that glucose concentration is less. It is possible that glucose affects the synthesis of cyclic AMP, which is formed from ATP by adenyl cyclase enzyme. So glucose की presence ही affect करती है cyclic AMP की formation को, and it is formed from the ATP by with the help of enzyme that is adenyl cyclase. This cyclic AMP is necessary for this cap binding proteins to bind to the promoter region. जो cyclic AMP है, it is very important. इसकी help से ही जो cap वाला portion है, this portion binds to this cap site. This cyclic AMP binds to cap, and once the cyclic AMP cap complex binds to promoter, the RNA polymerase can attach to the promoter and begins the transcription. जब ये cyclic AMP और cap दोनों ही bind करते हैं cap site में, after that this RNA polymerase will come to the promoter region and will start the formation of this mRNA, that is transcription. For example, in case of Lac Operon, when glucose level is high, even if lactose is present, the cyclic AMP is low, that we have already discussed, and therefore the cyclic AMP cap complex is not available to bind to the promoter and allow transcription to start. So अगर glucose ज़्यादा है तो cyclic AMP available नहीं होगा and it is unable to bind to this cap site, और जिसकी वजह से RNA polymerase transcription नहीं कर पाता. However, in the absence of glucose and in the presence of lactose, this cyclic AMP is plentiful. Cyclic AMP बहुत ज़्यादा होता है and is able to combine with the cap so that transcription proceeds. So if the glucose is not present, then this cyclic AMP is plentiful and is available to combine with the cap, and it will start the transcription process. The rate of Lac Operon transcription in the absence of glucose is 50 times as great as in the presence of glucose. So 50 times ज़्यादा होता है अगर glucose present नहीं है this Lac Operon का rate. This was all about for our today discussion about gene regulation, and in this lecture we have discussed about the Lac Operon in detail. Hope you will get some idea from this presentation. If you have any questions, queries and any suggestions, you can give it in comment section. Thanks for watching. Have a great day.